Hello everyone, I'm Alejandro Montenegro and I'm the executive editor for Cold Spring Harbor Protocols. I'm joined here today by Nicolas Plachta, who is a professor in the Department of Cell and Developmental Biology at UPenn, to talk a little bit about the research and the work they will present at the 87th Cold Spring Harbor Symposium, which this year focuses on stem cells. Thank you very much for taking the time today. So, work in your lab focuses on the early mammalian embryo, particularly on cellular organization. I was wondering if you could tell us a little bit about this and the main techniques and approaches you use in the lab to study these processes.
So yeah, the challenge for us has always been to look at the very early stages of embryonic development, and most of the information we had about how the mammalian embryo forms from the moment of fertilization until the cells divide many times and they start to differentiate and are ready to implant in the uterus. All that is called the pre-implantation stage of development. And it's very important to study this because many things go wrong in human embryos during that stage, and then we don't have implantation, we don't have a pregnancy. And the challenge was always for us to look at it in real time, how we can just move the field forward from just looking at, fixed specimens where we do histological analysis and look at real-time movies of these embryos where we can visualize cells dividing, interacting with each other, and forming the first structures, the first tissue organization in the intact embryo. So we've been coming up with technologies both for imaging and for culturing the embryos and manipulating them genetically to make different things fluorescent. So we can do that in real time. And it's been working well. Like we've been finding some things, and one of the interesting aspects when you do this kind of like live imaging research is that you find a lot of random things completely unexpected, which is hard to do with other areas or other approaches in research. If you work with biochemistry and genetics or many other disciplines in general, you need to have some sort of hypothesis, an idea of what you're doing. We don't so much. I cannot write a grant application to the NIH saying like, I'm just going to be imaging and maybe I find something. But in reality, most of the things we've been discovering are just random things via imaging.
So the focus on the early embryo is mostly because many things can go wrong, or because there's a particularly attractive thing about that system to study cell dynamics?
Both, yeah, excellent. So, pretty much anything you study that has to do with embryonic development is important. How the embryo forms, how the heart forms, how the brain forms, right? Just understanding how any piece of the body forms and how it functions is super important for understanding later how to fix it, what goes wrong. But it's true that the very, very early stages of development in mammalian embryos in human are just very fragile. We have a lot of chromosome segregation defects and aneuploidies. That means that the cells don't segregate very well the genetic contents during division, and then that can give rise to chromosomes lagging or extra chromosomes that can give rise to all sorts of, for example, Down syndrome and these kind of things. So, the main idea is that you start with a very unpolarized cell that just got fertilized. It's just like a one big round cell without where anything's possible, and it starts to divide into other smaller, boring-looking cells. But around 2 or 3 days of development, they need to start doing interesting things. They need to start polarizing, they need to start interacting with each other, they need to start differentiating into the first lineages or cell types of the embryo that will give rise to very different things, like the embryo proper, and then the placenta, all this tissue that surrounds the embryo, and is super important for early development. So it's the two things. It's very important to look at this process, and also it's just like a really nice system to study mammalian cells in intact conditions, because we really want to study how cells work, and especially mammalian cells, and especially in vivo systems, like in real tissues and organs. But it's really hard to do that with live imaging, because, with the exception of the skin, anything else you want to look using real-time imaging inside the body, even in a big growing embryo, is too deep inside the embryo. And the microscopy techniques are only that good once you start going too deep, you lose resolution, the cells are too tiny. So the embryo is a lot less challenging, and that's why we like it, because it's one big fat cell that divides into smaller cells. And during that early stage, it starts to do all those very important things: differentiation, polarization, the cells specialize into different things. But we're still talking about just a few cells, from 1 to 64, let's say. It's not hundreds or thousands of cells deep inside a very tightly packed tissue with other tissues on top. So the answer is, we also like the embryo because it's accessible.
Right. So your lab has, like you were mentioning part of this, has pioneered the use of various live imaging approaches to study the pre-implantation embryo in real time. So, and I think partly you've answered this, but why focus specifically on pre-implantation?
Because it's less challenging than post-implantation.
Oh, okay.
We should also be working in everything that happens after implantation. It's just much harder, because what I was saying, then the embryo is going to implant in the uterus. Mm-hmm. You need a uterus. It just doesn't implant on top of a tissue culture dish. And so all those things are inside the body and they're very inaccessible. The early embryo, what I forgot to mention, is that it's just like a fish embryo early on. We have the egg, it gets fertilized, it's floating in the oviducts and in the uterus. But if we take it out and we put it in a dish with very minimal growth conditions, mm-hmm. it does the same thing as when floating in the uterus. We're just floating in liquid until the time of implantation. And that's what makes all this possible and accessible, that we can get these embryos out, study them all the way until they're about to implant, and then just put them back in. And they develop in the very same way almost, with very small differences. Later on, you cannot remove an embryo from the uterus after it implanted and it has hundreds or thousands of cells. You're going to just destroy everything. Mm-hmm. And then it's hard. So then you need to start working with kind of like explanted tissues, and it's more messy, they die out, etc. So that's the main advantage.
Mm-hmm. So now moving into your talk this morning, so you focus on three main questions, which is how cells choose their fate, how they control their shape, and how they choose their position. So how are you approaching these separate questions?
I pitch it like this. The reality is that we just image lots of different things happening in the embryo, and we find randomly many times new structures or new mechanisms that help us to understand those questions, how they're choosing their fate, their shape, and their position. For example, there are many things we found randomly. We were looking at how the cells start to change shape and compact, and it was always thought that they just increase their adhesion with each other. And all of a sudden, we're imaging the embryos, and we find that they have these very long protrusions, like long arms, and they use them kind of like to hug each other, bring each other closer. They're philopodia protrusions. And then we understand, look at these things, that we think maybe they help the cells polarize. Let's manipulate them, let's cut them with lasers, or genetically ablate them. And then we can propose it's helping the cells to change shape. So that it's not that we were looking for the mechanism that would allow cells to change shape, and then we find those things. So many random discoveries that just happened like that. And then it helps us to understand what is it for.
Mm-hmm. So you also discuss the dynamics of the cytoskeleton in the developing embryo, and how something called asymmetric inheritance of these proteins could impact cell fate and other elements. I was wondering if you could tell us a bit about that.
Yeah, that's been one of the very big challenges in morphogenesis, developmental biology, cell biology. How cells sense what's happening to them, their interactions with other cells in a tissue, in an organ, and how that gets translated inside the cell nucleus to express these genes or those genes, or repress these or those genes. So the cell specializes into this or that lineage, right? And what we've been finding is that the cytoskeleton is very important for that. So because basically between the nucleus and the cell membrane, the cell cortex, and all those interactions, inside all that cytoplasm is the cytoskeleton. That's formed by different filament networks like actin, microtubules, intermediate filaments. And we've been having a lot of fun just labeling those things and trying to see, for example, in the latest work we've been doing, how the cytoskeleton enables the nucleus to be mechanically coupled via all those skeletal filament networks to the membrane, to the actin cortex. And basically we find that, as the embryo is compacting and some cells internalize and others stay on the outer surface of the embryo to form the future placenta, the ones that internalize will give rise to the actual fetus. And so one question was, how do they know when cells get in or stay out to turn on genes important for the fetus or for the placenta? So we find that the cytoskeleton allows that because it mechanically couples the cortex to the nucleus, and we, for example, start to measure how mechanical forces are transmitted throughout the cytoskeleton to impact the nuclear organization, the organization of the nuclear lamina and everything that's inside. For example, the mobility of transcription factors that turn on and off different genes or different chromatin structure states. So it's just studying the cytoskeleton is really good for us to understand how cells are changing shape, because all that's driven by the cytoskeleton, but also how information about their shape and their position can be relayed to the cell nucleus and to genes. And it's never a unidirectional thing. There's also things happening inside the nucleus that somehow impact the rest of the cell via the cytoskeleton. The second thing is because it's just beautiful. The cytoskeleton, when you label it, when you do live imaging of the cytoskeleton, it's just so pretty. All these different cytoskeletal networks. You can show it to just normal civilians who are not scientists. Hey, look at the cytoskeleton in a cell, in an intact animal. Say, oh, that looks very pretty. You don't even need to know, like, if I show you, I don't know, some biochemical study with some bands in a gel, that's not pretty. It may be super important, it's not pretty to look at.
So
it needs to be pretty, not only important. Yeah, the science we do has to look pretty, because we just like it like that.
You mentioned that one of the next steps in your research is moving to studying human embryos rather than mouse.
Yeah.
And you mentioned that one of the major limitations is that you can't, in most places, do any sort of genetic manipulation.
Right.
Tell us a little bit about how you've addressed that issue.
Yeah, so we're still working on those things, but the idea is that there are tons of human embryos which are donated for research because people do IVF, and at the end of their fertility treatment, you generate all these human embryos. Many of them get transferred back, and then we have babies born from IVF, and many others are never used because we produced them in excess, or maybe they were a little bit abnormal, and they never got transferred back in uterus, into the uterus. And so at the end of the treatment, you're asked, do you want to freeze them? Do you want to, for whatever reason, do you want to just discard them, or do you want to donate them for research? And in many instances, they are donated for research. Actually, there is tons of embryos donated for research in human IVF clinics all over the world. And in many countries and states, you can actually do research on them. The thing is that the embryos you get are not very early embryos. They're not at the 1 or 2 or 4 cell stage. It's closer to the time of implantation, at the blastocyst stage. And also, in many places, you cannot genetically manipulate them like we do regularly with mouse embryos to make things inside the cells of the embryo fluorescently labeled. So we can study all these fluorescent structures.
The beautiful structures.
Right, all the beautiful structures. So until now, we've only been using just normal light microscopes. But once the embryo gets too complex, it's really hard to see what's happening inside the embryo and all that. So one of the challenges in the future is to start finding ways to fluorescently label things in embryos without genetic manipulation. And for 2 reasons. The first one is just to understand how the human embryo forms, how it's different from the mouse. I'm sure it's going to be similar and different to different processes. And second, to maybe move some of those new technologies in the future to the clinic so we can help clinicians or embryologists choose which embryo should or shouldn't be transferred in uterus at the end of an IVF treatment. There may be many morphological parameters there that we just don't know yet, but if we could image them, they may be good indicators of embryo viability or the potential of the embryo to do well or not after you transfer it back inside the uterus.
So unfortunately, that's all the time we have today. I want to thank again Nicolas Plachta for being with us, and thank you all for listening.
Thank you.